to be with you today. So let's start. Before entering really into the core of our seminar, let's start to define the different topic that you would need actually to write whenever you are elaborating a manuscript or a research article. And you start with the introduction, right? So I'm talking about the body of the manuscript. So the introduction answers the question why. So why you choose that topic, why you would like to describe more or to investigate more regarding that topic. The introduction state a problem, and you also are proposing with the next sections the solution to that problem. In the introduction, you need to explain why the problem is important and review all the work that has been done so far in the attempt to solve that problem and to overcome the challenges that are related to that problem. So the introduction, as is stated, the word introduce the study by pointing out what is different about compared to past research. So which is the difference that make your study successfully considered for publication? Sometimes there are some challenges regarding the verbs with which you need to write the sentences. So, which is the tense in the introduction? The tense is a mixture of present and past. The past referring to the experimental procedure or to the discoveries that someone else made or you made before writing this manuscript. But the present is because the challenge is still standing, and this is the reason for which you are trying to address it with your manuscript. So past tense, therefore, when you are talking about something that is always true because it has been already discovered, it has been already identified and explained. And past tense is earlier research effort because it's something that it has been done in the past. And the present is to reinforce that the challenge is still there. So the problem is still there. So if we amplify a bit more, so the present tense is a fact or a practice that is generally accepted in the field. The past tense is the result of an individual experimental paper, finished events in the past with a specific time. The present perfect tense is a summary or generalization about the research in the field or general trend in society, no specific time or date, like for instance, has been done. And the present continuous or passive tense, it refers to an ongoing situation, is being developed, for instance. So here in this slide, you have the different tense that are more commonly found in manuscript writing. And then you can relate the verb tense with the different conditions that have been elucidated in this slide. There are 6 steps to provide an efficient and an effective introduction, not simply for you to describe your work, but also to the reader to read and become interesting in your field. The introduction needs to provide background information. It needs to set the content, what you are describing. So all the framework and the context related to the topic needs to be reported. You need to say why the topic is important and state it very clearly, because there is nobody that consider that topic more important than you if you want really to publish your article. You need to also mention and give credit to the past attempts that have been done to solve the problem or to answer that specific question. and identify the gap in knowledge that the paper fills. Because it's important for the reader to understand that that gap is you, that you are proposing the solution and how to fill it out. And then you can conclude the introduction by giving specific objectives of your research, just an highlight to maintain the interest of the reader in continuing going through your manuscript. Aim for about 400 words that are divided in three different paragraphs. And that's an estimate, right? You can increase the introduction in most of the manuscript, but try to be very efficient in the words that you are using. The structure of the introduction, you needed to begin with a general information, because this is how you set the content and the context. You frame it, okay, in a very general information. But then you narrow the focus to target the attention of the reader on the specific problem that you want to study. And then you state your study rationales and describe the aims that your manuscript will address. So if we look figuratively, is like an upside down pyramid, where I said you start very general, then you tackle the problem, and then you do the specific aims. What I mean for general background is something that is really setting the reader on the problem. If, for instance, you are writing about cancer research, for instance, if because your manuscript is about that, you might consider general some percentages or prevalences or ratio about morbidity and mortality in that specific context. And then you narrowed it down, drawing the reader to the point of the study. Then you pass to describe the methods, right? Which answer the question how. So you really need to take the reader through a logical sequence of experimental procedures that you adopted to find the results that we will discuss in a moment. You need to provide enough detail so that your study can be replicated. And I cannot stress this enough because it's very important. Science is something that belongs to all scientists that performs it. So you might want also to reproduce in your experimental settings, in your specific context, the same procedure that I'm using, for instance, somewhere else. So this is why it's very important that when I write my paper, I always detail the experimental procedure and the reagents, even providing references for you to compare, because as you know, there are also reagents that might be more specific than others. And this will allow other researchers to reproduce the results in an efficient manner, and then to follow exactly even updates in the protocols that you are detailing in your paper. But the tense in the method session is past because it's something that you already done. So begin writing the material and methods while you are performing the experiments. And I'm always advising students to do that because the method session is a very easy session to write. Whenever you write a protocol on your lab book, so then try to write it in the best possible way, inserting all information possible, um, carefully writing with some formalism that then you can easily adapt it to your manuscript elaboration. And like this, you will save time. But there is something that is not advisable for you to do while writing this session. Exclude some details, for instance, because what might not be important to you might be very important to someone else. And this is how Scientists keep ensuring the reproducibility of the research. Include any irrelevant information is not right to do that, because if it's irrelevant already to you, then is not something that is adding value, and therefore you don't do that. You also don't forget to include the published literature wherever is needed, because all your study is founded and grounded on specific scientific premises that are based on the discoveries of someone else. So you always need to cite the literature. And then do not present results in the method session or provide interpretation. Just describe the experimental procedure that you use. So, how you did that experiment, how you got that result. So the method should allow the duplication of the study, because as I said before, this is very important to all researchers. And this is why you need to include as many detail as possible, because other colleagues might want to repeat that. But also you want also to your methods to be known worldwide, because you might become a reference in your field by using that procedure that then many other scientists are using it around the world. So give always the source of material, make and model the equipment, the quantities and the duration of each time course or dose response that you are performing, the reason for which sometimes you might want to inject animals models if you are inducing diseases at night rather than in the morning, etc. Describe all the treatments, how you do it, and how you arrange for appropriate controls to be used, and then refer the results that you got. So, like this, you have valid comparison between different data sets, and it's easy then to it's easy, but it's also mandatory to be able to compare the experimental results with the proper control. And this is the easy session to write, as I said. So it's a good idea to start writing this session, because it's just a reformulation in a more formal way with the defined word limits, etc., of something that you already wrote. So it's really to take back what you've wrote and just adapt it to be then inserted into this manuscript. What to include? Enough detail. Use the figures that sometimes you might add for clarity. What I intend for it. Imagine that you are describing a methodology, a strategy to identify specific populations, immunocells, with flow cytometry. But then you are just exposing then the results in the result session, right? And all the strategy, instead of describing it, you can prepare a figure and said, I did it this way. And like this, it's easy also for the reader to replicate the gates, to replicate all the procedures that you use. This is how you use figure for clarity. Sometimes I have to say, you might also use tables for clarity. Imagine that you are describing the genetic expression of specific samples, and then you are using a lot of primers to do that. Instead of writing it, all those primers in a text, so then you can use a table and put it all together. So it's something that it's improving clarity, and provide all details that does not fit the body of the manuscript in the supplemental data, because like this, as I said, everybody can reproduce it. It is important to state the funding sources of, for instance, the reagents as well, or the ethical compliance. So the institutional review board guidelines as needed if you are using also animal model or human samples. The verb tense is past because you are describing a procedure that you already adopted. The common voice is the essay was performed, and this is an example. But also the active voice is acceptable, like we performed, we evaluated, we developed. And I wanted to highlight actually this we, because in science, there is not me and you, is we collectively, because we achieve a result just if we join forces and collectively we produce what we aim for. There are essential checks for this session, like to be specific and detailed as possible to facilitate reproducibility, to give information on design, samples, selection criteria, ethics, trial registration, and consent detail where applicable, and procedures as well. To use diagrams or flowcharts to facilitate the description of the procedure if necessary, for instance, for design, but then include also data analysis session to explain the choice of the statistical test or the analysis test that you are using to really make and to draft the conclusion of your manuscript. And this is very, very important because most of the people drawn very different conclusion according to the statistical test. So statistics is not something that you want to use not in the right way, because it can give you potentially an interpretation of the results that is not what is correct. So pay attention also to this session. Action and phrases in the method session. So you need, as I said, to contextualize the study methods and provide the background information about the study. And these you need to situate your choice for the methodology in relation to previous study. And you can reference this methodology by saying X was prepared according to the procedure used by, or you might want to provide the relevant theoretical or empirical information to show connections to the field, like traditionally, X has been assessed by measuring, or you want to reinstate the research purposes, hypotheses, or gaps in research by saying to test our primary hypothesis that, or name the methodological approach. The design was a mixed method study, or describe the physical setting of the study, like, for instance, observations were conducted at X hospital, or introduce the subjects and participants, like criteria for selecting the subject were as follows, or to justify pre-experiment choice, like a major advantage of X is that. But you also want to describe the study in detail, because these move typically between the 55 and 85% of the entire method session if the details are well provided. So you can explain how the data were collected, sampled, or selected by saying was recruited, for instance, this sample of patients. Describe the characteristics of the data by saying data consists of, or identify independent and dependent variables by saying our independent variable is, or describe the study procedure in a step-by-step way. The first step in this process was the soil was then weighted again, the resulting solution was gently mixed, minimum frequency was set to. And then you might also want to analyze data to show exactly which step you took to persuade the reader that the results can be trusted, like describing tools used in the study, like the data management analysis were performed using, and then you put the software and the license, or to share observation or incremental measurements related to the study but not crucial to the results. You can say explain how data were prepared for the analysis by saying the data were normalized, or you might want to describe how data were analyzed, including coding schemes or tool. There might be an overlap with tooling of tool, like the number were entered into, or by justifying data processes as analysis, saying this framework was chosen because, etc. So there are some papers that you really need to check the instruction guidelines for authors, because the meta session might be before or after the results. If it's before, is follow straight to the introduction, then there is the meta session, the results, and the discussion. But there are some journals that prefer to continue capturing the attention to of the reader until the end, and then explain how the results were obtained by introducing the for the meta session at the end. And it varies by journal and journal. In Nature, for instance, the method is in a smaller font at the end of the paper, because what is important for the journal to highlight is the take-home message. And now we are coming to the core of our webinar, which is the results and the discussion. You need to state the results of the experiment, describing a possible explanation for the results, and direct the reader to figures and tables that present supporting data. My advice is to get all the results you would like to publish, and then try to see how you define the story in the best possible way before even start writing it. The results answer the question, what? So what? So what did you discover? You need to list the results and describe the tables and the figure. You need to make sure that you cite all the figures and the table in the text, because sometimes it is not done, and then there are always reviewers that can point it out. And then provide the numbers and any statistical values to imply the significance or lack of significance. Do not forget that. You can put the p-value on the graph indeed itself or in the legend, or and put a star, for instance, in the graph, or 2 or 3 according to the significant level of your results. The tense in the result session is past tense. So how you effectively present the results? You state only the results. So if you have a graph in front of you, you just analyze the graph by explaining what is the result that the graph is showing to the reader. Don't insert any comment, but leave all comment and the explanation, meaning the interpretation of this graph, for the discussion session. Just limit yourself to describe what are the major findings and answer all the points that has been raised in the method session. Use tables and charts as appropriate, but do not duplicate the data by presenting the same data once as a table and once as a graph, or repeating the graphical data in the text. I open a parenthesis over here. Sometimes it's useful to confirm the data with another methodological procedure. So you might show that even in the supplementary figure if for you is really important. But duplication of the data, it does not add value to your text. So no mismatch in numbers between text and tables or figure, and this again, pay very much attention to it. So all this session can be shortened of the IMRAD session, because a lot of information can be presented in tables or figures, and they really needed to be self-explanatory. So group the results chronologically, presenting the results in the order in which the experiment were performed will make it easier to relate the results to the methods. But sometimes you might start in a way and then you add more experiment to confirm your hypothesis, and therefore you cannot sell the story, let's say like this, in a chronologic manner. Then you can do it by topic. So you present the results by topic that make easier the reader to possibly understand it. And this is an example, like the performance of a method or a material or a device in comparison to others. So there are specific settings on when to use tables and when to use figures. So if you are indecisive, these couple of slides will help you out, because you can use a table to show numerous values or other data in a small space. Sometimes you use a table to compress all the data that you might have, that otherwise you would need 50 pages just to provide the graphs about it. And you use a table also to compare values or characteristics among the groups, or to show the presence or absence of specific features. But you use a figure to show trends, to show patterns and relationship in data, when the general pattern is more important than the exact value. If you want to see an increase, for instance, right? So then you can use a figure reporting graphs as histograms or others, or to illustrate a sequence of event, procedures, geographical features, or physical characteristics. So the figures, remember that needs to be informative and self-explanatory. So figures offer readers a quick overview of the major study findings. The figures help the readers to better understand the study results. And the figure should have a brief description or a legend, providing the reader sufficient information to know how the data were produced. Remember also to look at the guidelines for author's instruction, because you need to prepare also the figures according to the recommendation of the journal itself, in terms of how many figures you can prepare, or the resolution of each figure is something that you need to pay very much attention, as well as the format that then you will need to upload in the journal platform. However, whenever then you start preparing the figure, you really need also to pay attention to the colors. And there are some basic principles for colors use. The tone color should be consistent throughout the paper and not mix the color according to the figures that you are presented. I'm making an example here. For instance, if you are uh, setting the control in an histogram that has a black border but is white, to what concern the filling of the histogram, always make the controls white. And then you can make the treatment. So you compare the treatment results to the control. You can do it black. Okay. And then you continue to do the treatment in black. What is the benefit for it? Is the benefit is for the reader to easily spot your data without even reading entirely the manuscript. And remember that you might have multiple conditions for which you also need to use gradients. And you can do it like darker colors, increase numbers, but lighter colors decrease numbers in the gradient. But it's important though that you don't fill or make graphs with lighter colors, because most of the time the lighter color is poorly visible, even when printed or when it's online on the journal. So try also to make the decision in a way that even if sometimes after your manuscript publication present the data the people in the back can see the color. And highlight important parts. For instance, if you want to use the red, you can do it, like in this example. The knockout data of this trial is in red because it is more visible and is more easy to see the difference when compared to wild type animals. This is also another example of an immunostaining of RGC neurons. And then you can see the different proteins that are stained either in red or in green, but there is the use of the errors that point specific patterns that you would like to highlight, and then you insert the scale. So the more that you read, the more you can see also how results are presented, and then you can adapt yours in the best possible way. Avoid all unnecessary figure like this one, right? This is a really unnecessary. You can simply state it in the session, in the description of the results, that in this case, 63.4% were women. You don't need to provide a figure of that. If you are also using age groups, you don't need to provide the figures for age group. It's just a sentence, and you avoid unnecessary measurements. So, important point for table and figure. You need to include the titles and the legend as necessary. Make the legend short and precise. The figure and the table should be able to stand alone and never present the same data in tables and figures. Also, when you write the results, remember not to exclude the negative results. The negative results is very important for someone else on top of yourself not to take specific directions for the project that you already tested. But it's also important for you to highlight and emphasize the positive results. So do not report every results you obtained because it's a manuscript, it's not an encyclopedia. Remember that, contrary to the expectation of early career researcher or students, is less than 5% of what you produced in a PhD program that you publish. Don't be tempted to include numerous not textual elements. Do not interpret the results. You will leave these for the discussion, and do not use references because are your results. Instead, include only results that are pertinent to the question that was posed, that report both positive and negative results, and that summarize the data that you obtained along of your scientific training. As essential check, state the most important results first, and then try to build up the story according to that and to explain that. Use journal prescribed format for reporting results and statistics. Use tables like when providing data characteristics, reporting full results for extensive comparison, like p-value or confidence intervals or effect size, etc. Use figure to highlight the significant findings and key results that will be then discussed further, and provide the table and figure legend to ensure that they are self-sufficient for interpretation. So this session is the shortest of the body of the manuscript called the IMRAD, because a lot of the information can be presented indeed in tables or figures. So you need to use the results to make the reference to the aim, to the research purposes and the procedure, to make, and like this you can, to identify the difference between A and B, for instance, if you want to write a sentence, or To make the reference to data or statistics, referring the data in table or charts, or emphasizing significance data trend in table of chart, and you can say figures 1 compare or figure 10 demonstrate. To evaluate the result, stating a positive result or stating a negative result, or highlighting interesting or surprising results. Strong evidence X was found. There was no evidence that, interestingly, X was observed. Reporting survey and interview findings like response rates, proportion, teams, participant view experts. So respondents were asked to indicate whether 70% of those who were interviewed, another reported problem was, one interviewed argued that. Or to make transition, seamlessly moving out of one result into another, or summarizing, like comparing the two results, it can be seen that, or overall the experiment showed that. And these are examples, right, of how to write it. For instance, in the adjusted multivariable model, several of these associations persisted, and then you insert the table for people when go direct the reader to see the table. Teaching hospital had higher CIT scores, etc. Or we found no relationship between the normalized IT expenditure, etc., indicating that, and then you put the sentence diminished after accounting for bed size. So like this, you explain the results that you got. You do not interpret it. Again, another example, we did not observe a trend, etc. Or from 2004 to 2010, the proportion of E. coli, etc., rose from 5% to 23%. And then you insert the figure for the reader, go into the figures and look at it. So the results are basically claims that can be classified accordingly, like basic claims: an increase in smoking among teenagers cause long-term health problem, or stronger claims, like based on these results, it has become clear that an increase in teen smoking must have caused limited claims. We have reason to believe that, or you can also say in the best of our knowledge, right? So, and then you continue. The discussion. The discussion follows the results and answer to the question, what does it mean? And what did you learn from this study? And provide a sort of larger picture first, but also include the limitations of the study and the future directions. And why I'm literally showing you the limitation before the future directions, because most of the direction that you might want to explore in the future are because of limitations of the study. So those are very important to be highlighted. For the tense, you use the past tense, the present perfect, to summarize findings, but you use the present tense to interpret the results, because this is your interpretation, your significant discussion, and your conclusion presentation. And then you use the future tense to make recommendation for further research, or to indicate a future course of action based on the results of your paper. So it is important that you state the clear answer to the question that you posed in the previous session. For instance, we tested if migratory bats, etc., respond to artificial green light at night during migration. Or our study is the first to show that migratory bats responds, etc. So you state it, it's clear. But you also give support and explanation to defend those results. Our estimate mortality is higher than, probably because, or the high proportion of E. coli, bacteremia, etc., are consistent with. So you explain, and you really make an argument to defend the data. And you establish the limitation, the implication, and the relevance of your findings. our study highlights the need, or our study confirmed the importance, or what we observe in these results suggest that, or a limitation of the study is, etc. So it's something that it closes the context that you open up at the beginning. And this is indeed how is the discussion. The discussion is really contrary to the introduction structure, meaning that the discussion begins with a statement of your main findings. So is beginning in a very narrow manner, and then it broadens, because then you really need to open up the topic to generalize it at the very end. So it's exactly the opposite. How you write an effective discussion with the implication, so the theoretical implication that you might want to add to all the existing literature and theories, or the basis for new theories, while practical implications are important ramification for practice. But you also wanted to write a recommendation, there's some suggestion to propose, or proposals about something that should be done, like to improve the study or the methodology, or to suggest policy or a direction for future research. So it is important, therefore, and I really wanted to express the aims of the discussion, state a clear answer to the question, support, explain, and defend the results, and establish limitations, suggestion for the future implication and relevance of your finding. Do not write another introduction. You don't need that. It's different, it's an interpretation of the results in the context of the problem that you describe in the introduction. You don't repeat the results or introduce new findings, you interpret them. You don't exaggerate or overgeneralize the results. You don't criticize our other studies. You don't forget to mention the limitation and strength, and you don't forget to give the manuscript a sense of completion, indicating opportunities for future colleagues to conclude with clinical or commercial implication, if any. You need to close the story with the discussion. And this is why you need to really explain what the result mean and why are so important. Compare also the results with early findings to explain contradictions, if any. If some results did not attain statistical significance, explain any differences seen might have been due to chance. Explain how the researchers move the body of the scientific knowledge forward, why, therefore, needs to be accepted for publication, but also outline the limitation of the study and suggest the future direction, and then sum up with a conclusion. But do not extend your conclusion beyond what is directly supported by your result, because you need really to avoid undue speculation, because otherwise it's more an interpretation paper or an opinion that you can write also, but is a different topic. So the style for the discussion session, the results, the discussion, and the conclusion might be either separated or combined according to some journals. And for instance, the science journal are mainly combined. Review the target journal style and prepare your manuscript according to the instruction of authors for authors, and then include always the take-home message. So regarding the action, remember to provide the background information, references the literature or the question prior study have of with respect to the first research question, or reinstating, comparing, and explaining the results, summarizing the results, supporting previous finding, contradicting the previous findings, indicating an expected outcome, and offering explanation for findings. And here you have some sentence examples, but also advising caution interpretation of findings, like although exclusion of X did not, etc., and suggesting a general hypothesis it is therefore likely that connection exists between, etc. And you can find more about the common moves in the discussion session in the link that has been provided in the slide. As essential check, discuss the results in the context of the research aim and previous literature, acknowledge conflicting or unexpected results, do not simply repeat the results, do not exaggerate or overgeneralize, do not forget to mention limitation and strength, and suggest implication and new hypotheses to warrant further investigations. You finalize then your manuscript with the conclusion that, as I said, can be two sessions separate or combined according to the journal. You can provide in the conclusion, actually, you should provide a take home message and mentioning the strength and the limitation of the study in a brief manner, and also indicating a short and long term implication. But remember, this is a short session. And it is important that you always have in mind that you need to use what we call the ABC of science writing for this session. Indeed, the brevity, make things as simple as possible, delete unnecessary word from title and abstract, but also from discussion, from results and methodology, considering the importance of the length of every paragraph and sentence, and evaluate the need for the presentation of every table and figure. And in terms of clarity, try to think as a reader, capturing your own attention, and identify and eliminate every ambiguity that your manuscript might have, focusing also on clarity at the level of word, sentence, paragraph, and the overall text. As Marcus Fabius Quintilianus was saying, clear writing is writing that cannot be misunderstood. And with that, I really would like to thank you all for your attention. My name is Raffaella Gozzolino. I will now stop sharing the slide, and I will be very happy to respond to any doubt or question that you might want me to clarify. Thank you very much.